Hi, Michelle Glass here. Welcome back to another one of our Chapter 22 Part 2 lecture series. Our topic here is the discussion of those Cd4 T cells, how they get activated, and also activation of our B cells. Now we will be looking at them side by side. So we're going to see what's happening on the left side of the screen first. That's going to be our Cd4 T cell activation, and then on the other side of the screen we're going to be seeing our B cell activation. I did represent them side by side because both of these things are happening simultaneously in the body. Keep in mind too that everything we've already talked about with CD8 T cells should also be happening simultaneously in the body, in addition to all those non-specific defenses that we've discussed in Unit 2. Alrighty, so let's begin by looking at our APC, or antigen presenting cell. Remember, they're going to be either microphages or macrophages. They're part of our non-specific defenses. They're moving through the body, identifying foreign pathogens, and then phagocytizing those. B cells are also doing phagocytosis, but they are part of our specific defenses. So they are identifying specific antigens, phagocytizing those. They have, as part of their cell membrane, a special group of antibodies or immunoglobulins called IgDs. And what you're looking at there is an inactive B cell. It's inactive, but it is doing phagocytosis. Now remember, our immunoglobulins, our antibodies, our IgD molecule will be specific for specific antigens. Okay, taking a look at our antigen presenting cell, it is going to start out an immune response by presenting an antigen. Now remember that macrophage is non-specific defense. So even though it knew that that bacterium was foreign, it doesn't know who it is. It's just hanging off that antigen as a flag using its class 2 MHC proteins to initiate the immune response. And then we see our B cell, remember, did phagocytosis of that same antigen, and previously it was inactive, and now we call it sensitized. Two things have to happen for it to be sensitized. It has to present the antigen using class 2 MHC protein markers, and it has to have antigen bound to its IgD antibody. Once both of those steps have occurred, that B cell is called sensitized. So clearly, if you have this pathogen in your body, you would expect to have macrophages engulfing and presenting those antigens, and you would expect to have B cells becoming sensitized. Okay, the B cell sensitization is going to be, how it will be for a little bit of time. So we're going to kind of leave that one alone and focus in on getting that CD4 T cell activated. So we've already done antigen presentation. So of course our next step is recognition and binding. So because we're holding out that marker with a class, or that antigen with a class 2 MHC marker, it's going to be a CD4 T cell that recognizes and binds. Even though the macrophage doesn't know who is holding out, it just knows it's foreign, that CD4 T cell is specific, so it is only able to recognize that particular antigen. So just like we had millions of CD8 T cells that can recognize and bind to millions of possible antigens, we have millions of CD4 T cells that can recognize and bind to millions of different antigens. You should know our next step. You're expecting to see co-stimulation. So again, focusing in on what's happening with that CD4 T cell, we see that the macrophage does the co-stimulation, which remember is like, letting go of the safety on the gun. It is that confirmation for that CD4 T cell to go ahead and activate. And once that CD4 T cell activates, then it's going to divide, and it's going to be making two types of cells. We're going to get a helper T cell and then memory helper T cells. Remember, those memory cells are inactive that first time. We're going to see them being involved in subsequent antigen exposures. It's in this first exposure, it's that helper T cell that's going to do all the work. So helper T cells are releasing, again, chemicals in that group called cytokines. We will see that these cytokines are going to have multiple effects. Number one, we're going to see them increasing our cytotoxic T cell maturation. So remember I said, if you have this antigen present, not only are you going to activate your CD4 T cells, not only are you sensitizing your B cell, but you're also going to activate your CD8 T cells, which gives us our cytotoxic T cells. We also see that these cytokines will attract more macrophages. So that gives us, more pathogens being removed. That gives us more possible antigen-presenting cells. We also will see that these cytokines are going to attract those cytotoxic T cells to this area. So we're bringing all these defense cells and speeding up their maturation and so forth. And then we're going to see that the helper T cell is going to help get that B cell activated. This whole time, our B cell has been sensitized, and that's it. So now we see that helper T cell that we've just made will recognize and bind to our sensitized B cell. Note that the B cell has to be sensitized before you can have your helper T recognizing and binding. Then that helper T cell is going to release a cytokine, which is co-stimulating the B cell. So it's kind of like the helper T is saying, and maybe I should have reversed my little cartoon expressions here. The B cell should be saying like, should I get busy? And then the helper T cell is saying yes. So the helper T is co-stimulating with the release of cytokines the B cell. The B cell then activates and divides, and it's going to make a memory B cell and a plasma cell. That plasma cell, remember, is the cell that releases antibodies. These antibodies are specific to this particular antigen, and we're going to be looking in a whole other video about the jobs of these antibodies. Plasma cells can release up to 100 million antibodies in a single hour. So, wow, that is a lot of antibodies. This is, remember, part of our humoral mediated defense. So this is going to be released into body fluids. You can think of them right now as acting like smart bombs. So they're going to actually, or like a targeted drone strike, they're going to actually be able to find these specific antigens, and then they have a whole list of things they can do to help as part of our defense. Okay, so stay tuned, but that's it for now.